Hi, welcome to Raaonline. I am Dr. Ashok, Assistant Professor of Microbiology. So today we are going to learn an important infection which is pertaining to the central nervous system. So these are the particular pathogen which belong to the parasite, and it is going to be an protozoa which is going to cause an extensive damage to the central nervous system. So we are going to deal with the free living amoeba, especially the Naegleria fowleri, which is going to cause an extensive tissue damage which is going to involve the central nervous system. So let us go into today's topic. So what is a free living amoeba? So free living amoeba are the amoeba which is distributed in the soil and water and which can cause opportunistic infection in the human being. So when we talk about free living amoeba, there are four important free living amoeba which is going to cause damage to the human, and those four important free living amoeba are Naegleria fowleri, Acanthamoeba species, Balamutaya mandaralis, and Sapnia. So in this, Naegleria fowleri is going to cause something called primary amoebic meningoencephalitis, and Acanthamoeba, they are going to cause Acanthamoeba keratitis and granulomatous amoebic encephalitis. And again, Balamutaya species, so they are going to cause something called granulomatous amoebic encephalitis. And Sapnia, again, it's going to cause a varying form of encephalitis. So these are the particular pathogen which belongs to the protozoa, and they are going to involve the central nervous system and cause wide range of manifestation. So most of these species are going to cause life-threatening infection, and most of the time the risk factor is going to be the water source like a pond and river. So anything like pond or river which is going to be an unclean environment, so they are going to harbor this particular pathogen. So whenever we are going to have a bath in those kind of water, we are going to acquire this infection which is going to cause a life-threatening condition. So with this, we are moving on to the Naegleria fowleri. So today we will deal with the Naegleria fowleri, and it is going to be a free living amoeba which is going to cause something called primary amoebic meningoencephalitis. So this particular thing can be asked in competitive exam. So there are four species of free living amoeba which is going to cause what kind of illness. So in case if it is going to be Naegleria fowleri, it is going to cause something called PAM, which is called as primary amoebic meningoencephalitis. So again, the risk factor is going to be the anything swimming in contaminated water a pond or river which is going to be contaminated with the trophocyte form of this particular pathogen. So we are going to be at a high risk of harboring this infection. And less immunity. So this particular primary amoebic encephalitis, so it can occur in any age group and even in healthy individual, but people with low immunity, they are prone for high rate of or high chance of complicated form of infection. So most of the time it is going to be our cell mediated immunity which is going to take care of these kind of pathogen. Again, when our cell mediated immunity is going to get impaired, so we can end up in these kind of infections. So this is not only going to attack the immunocompromised individual but also the healthy individual. So in India, the cases have been more than 20 cases have been reported, and the cases have been reported from areas like Mangalore, Calcutta and Karnataka. And when we study about the mortality rate is very high for this particular infection. It is going to be around 98%. So usually death occurs within 7 to 14 days of diagnosis. So again, it is very important to know the percentage. So the mortality is going to be 98% for Naegleria fowleri, primary amoebic meningoencephalitis. And coming to the morphological form, so when we talk about this particular species called Naegleria fowleri, which is a free living amoeba, so there are three forms. One is going to be the amoeboid form, flagellated trophocyte and cyst. So again, there are this amoeboid and flagellated form. So those both are a trophocyte form, and that is going to be a double-walled cyst. So in case if you are going to see about the amoeboid trophocyte form, this amoebic trophocyte form is going to have something called lobopodia. So this is called the lobopodia, which is a motility organ. So with the help of this lobopodia, this particular organism is going to move from one place to another. Again, this is called as the amoeboid form. So hence it's going to contain this lobopodia. So due to the presence of this lobopodia, we are going to call this trophocyte form as an amoeboid form. Next, we are going into the flagellated form. So here, what is going to happen is that this particular lobopodia is not here. So instead of lobopodia, it is going to contain something called flagella, which is going to be the locomotory organ. So with the help of this particular flagella, the organism is going to move from one place to another. So this is about the flagellated trophocyte. And another form is going to be the cyst. So whenever there is going to be an unfavorable environment for this particular trophocyte form, so what is going to happen is this trophocyte will become something called cyst. So the specific morphological form of this particular cyst is going to be the double layered cyst. If you closely see, there is going to be a double layer. So this is going to be a classical example for an cyst which is going to be seen in Naegleria fowleri. So again, we are going to see about three morphological forms in Naegleria fowleri. One is going to be the amoeboid trophocyte form, then flagellated trophocyte form, and then we have the cyst which is going to have a double wall. Now moving on to the etiopathogenesis. So whenever like we are going to have a bath in the contaminated water, like contaminated river water or any pond water, so we are going to come in contact with the particular pathogen. So what is going to happen is that the trophocyte form is going to come in contact with the nasal mucosa. So this is the primary step in the infection. So this particular trophocyte is going to come in contact with the nasal mucosa. In the nasal mucosa, like it is going to produce various enzymes like cytolysin and phospholipase. So with the help of that, it is going to penetrate the nasal mucosa and it is going to reach the olfactory nerve. So it is going to penetrate the nasal mucosa. So with the help of virulence factor like phospholipase and cytolysin. So after that, it is going to involve the olfactory nerve, and after reaching the olfactory nerve, it is going to travel to the brain. So in the brain, it is going to cause something called primary amoebic meningoencephalitis, where with the help of amoebostome, it is going to form something called as a food cup. So with the help of that food cup, it is going to digest all the brain tissue and it is going to engulf that brain tissue, which is going to serve as an energy for that particular amoeba. So it is going to utilize our brain tissue as a food for the particular parasite. And following that, is going to be death. So you all know like the mortality rate for this particular infection is going to be around 98%. So once we are going to get this infection, then mortality rate is going to be 98%. So if you closely see, there is going to be a cyst stage and there is going to be the trophocyte form, which is amoeboid form, and there is going to be the flagellated stage. So we have studied in the previous slide that so the morphological form of this particular pathogen is of three types. One is going to be the two trophocyte form and one is going to be the double-walled cyst. Again, the trophocyte form is going to be the amoeboid trophocyte form, which is going to contain the lobopodia, and another trophocyte form is going to contain the flagella. So both of them going to move with the help of the lobopodia and flagella. And third form, when the condition becomes unfavorable, like it is going to become a cyst, so the cyst is going to have a double-walled cyst. Now moving on to the clinical feature. The clinical feature comprises of fever, nasal discharge, feature of meningitis and neuropsychiatric manifestation. So whenever like we are going to deal with an infection or any pathogen, there is going to be an increase in temperature, body temperature. So here again the patient is going to have a fever. It could be either a continuous or intermittent type of fever. So most of the time it's going to be a high grade fever, hence it's going to cause an invasive infection of central nervous system, and there is going to be a nasal discharge. So here like what is going to happen is that during the primary invasion, it is going to affect the nasal mucosa, where it is going to produce certain enzymes like phospholipase. So again, like these kind of enzyme, they are going to have an damage of the nasal mucosa. So due to the particular thing, the patient is going to experience something called rhinorrhea. So where the patient is going to have severe nasal secretions. And following that, the patient is going to go for the stage of meningitis. So during the meningitis, like patient can be having variety of symptoms. The patient can be having a blurred vision, photophobia. The patient can be having an altered sensorium, and the patient can also have a severe headache. And when they are going to come to us, like we are going to elicit the sign. So at that particular time, the patient can be having something like positive nuchal rigidity sign, and Brudzinski sign may be positive, and Kernig sign can be positive, which is going to tell us if the patient is going to have something like meningitis or encephalitis. So most of the time it's going to cause encephalitis, but still it is going to have symptom which is going to be looking like moreover of meningitis. So last thing is going to be the neuropsychiatric manifestation, where the patient is going to experience something like an delirium or coma. So here like patient can have varying type of illness, starting from fever to the neuropsychiatric manifestation, which is going to tell us if the patient is going to having any illness which is suggestive of this kind of meningeal involvement. So this particular clinical feature can tell us if the patient is going to have a simple illness or going to have an illness which is going to involve the central nervous system. So with this, like we are moving on to the radio diagnosis. So once we are going to get a patient who is going to have this kind of illness, the patient is going to have certain symptom which is going to be like more suggestive of infection of central nervous system. So we will be having a non-invasive radiological investigation like CT or MRI. So in case of this kind of amoeboid invasion, so the patient can have some findings like obliteration of assistance, and again like the patient can have an leptomeningeal enhancement, and diffuse enhancement can be seen around the midbrain. So these are the classical feature which can be seen in case of primary amoebic meningoencephalitis. Now moving on to the lab diagnosis. So again like now we have seen a patient like who is going to have signs and symptoms of meningitis. So with the signs and symptom, we have underwent an procedure, that is a non-invasive procedure, that is a CT or MRI, which showed certain signs of leptomeningeal enhancement or anything like which is going to be confined to the central nervous system. So with this finding, like we are going to go for the next invasive procedure, that is a lumbar puncture. So with the help of lumbar puncture, like we are going to aspirate the CSF specimen and we are going to look it under the microscope. So the first thing which we do is the CSF wet mount examination whenever we are going to have a suspicion of amoebic involvement. So in case of amoebic involvement, like we will be able to see something like an trophocyte or even the cyst form. So here like we are going to obtain the CSF specimen, and immediately like we are going to do an wet mount examination. So here like we will be able to see the presence of trophocyte or the cystiform. And the thing about CSF is that like when we are going to suspect something like amoebic involvement, like the particular CSF should not be refrigerated. In case if you are accidentally going to refrigerate that particular CSF specimen, the particular architecture of the amoeboid form can get destroyed. So hence we are not supposed to refrigerate that particular CSF specimen. And in case if the patient is going to die, like the mortality rate is going to be very high. It's going to be 98% for this particular patient. So the mortality is going to be around 98%. So if the patient dies, like we can go for post-mortem examination. So again, like, so we can take the biopsy specimen from the brain, like where we can stain the particular histopathological section with the hematoxylin and eosin stain or any Romanowski stain like GIMSA. So in that, like we'll be able to see the nucleated amoeboid form. So it can tell us the cause of death in certain doubtful cases. And something like we are moving into the culture. So again, like it is going to be an parasite. So this particular parasite can be cultured. So we use something called non-nutrient agar, which is going to contain the page saline. So the particular parasite grows well in this particular culture medium. And we do something called N-flagellation test. Now moving on to the isoenzyme analysis. So we do something called isoenzyme analysis, like where we are going to analyze the particular CSF specimen for isoenzymes like malic enzyme and hexokinase. So this will give us an idea about the presence of a particular amoebic pathogen, and it will also help us in differentiating like what kind of amoebic pathogen it is. And we do the molecular test, so that's the PCR, so where we determine the presence of 18S rRNA, which is going to indicate the presence of a parasitic etiology. Now moving on to the CSF wet mount examination. So where we are going to do the lumbar puncture, and we are going to aspirate the CSF specimen, and we are going to do a wet mount. So in the wet mount, like if you are closely seen, like you are able to see the amoeboid form, and you are going to see the lobopodia. So these are the trophocyte amoeboid form with the lobopodia. And here like the particular trophocyte can move from one place to another. So it is going to be an amoeboid form where there is going to be the lobopodia. And with the help of this particular lobopodia, this particular organism is moved from one place to another. So there is going to be a live form. And in certain case, like you will be able to see something called cyst form. So again, like the cyst of Naegleria fowleri, it is going to be the double-walled cyst. So where you will be able to see a double-walled cyst in the CSF specimen. So when anything like unfavorable condition, so in the presence of unfavorable condition, this trophocyte is going to become something called a cyst form, where you will be able to demonstrate the particular cyst structure in the CSF specimen. Now moving on to the GIMSA stain. So when we are going to have a suspicion of this kind of free living amoeba infection, like we are going to subject that particular CSF specimen for special staining method like Romanowski stain. So one of the most common Romanowskis which we perform in the CSF specimen is going to be our GIMSA stain. So here like you will be able to see the Naegleria fowleri along with the pus cell. So if you closely see, there is going to be the neutrophil. So these are the neutrophil. So and these are going to be the particular pathogen, that is the free living amoeba. So here like you are able to see the nucleus, and again like you are able to see the cytoplasmic vacuole. So these are the pathogen. So these are the pathogenic part which is going to suggest the presence of free living amoeba like Naegleria fowleri in the specimen like CSF. So again like it is going to be a special stain, like where we are going to study about the particular parasitic morphology. So here the black arrowed thing is going to be the parasite, that is the trophocyte of free living amoeba, and again like this white arrow, it's going to indicate the presence of neutrophil, which is the pus cell. And this is a histopathological section which is going to demonstrate an brain biopsy. So this is a brain biopsy specimen which has been stained with something like GIMSA stain or hematoxylin and eosin stain. So here like you are able to see a nucleated organism. This is a cytoplasm and this is going to be the nucleus. So again like this is a classical example for histopathological section which is going to demonstrate the presence of the pathogen inside the histopathological section. So again like it is going to be an biopsy specimen. In case if the patient have died and we want to know the exact etiology, we are going to perform something called biopsy. So we are going to remove the patient's brain out, and we are going to do a section, and we are going to study, and we are going to do some special staining where we are going to analyze the particular section for the presence of anything like this free living amoeba. So here like you are able to see a basophilic structure which is going to have so here you are able to see a basophilic structure with the nucleus. So it is going to indicate that the patient is going to suffer from the infection of free living amoeba. So now moving on to the various specimen section. So here like you are able to see 6 images. So the images 1. So the image A, B, C, D is going to be a radiological investigation which is going to tell us about the radiological finding which is pertaining to the amoebic lesion in the brain. and image E is going to be the gross specimen. So this is going to be the gross specimen which is going to show us the presence of hemorrhagic necrotic lesion which is going to be caused by these kind of free living amoeba. So these kind of free living amoeba, they are going to produce something called amoebostome or foot cup. So again, with the help of this foot cup, it is going to erode the particular brain parenchyma and it is going to cause hemorrhagic or necrotic lesion. So this is about the image E. So now moving on to the image number F. So the image F is going to demonstrate the presence of histopathological section which is stained with the hematoxylin and eosin stain. And if you closely see, you are able to see a cytoplasmic thing, that is the eosinophilic cytoplasmic pathogen which is surrounded by the inflammatory cell. So these are the inflammatory cell, and you are also able to see the particular pathogen. So this is the eosinophilic cytoplasm of the trophocyte of this particular free living amoeba. So again, like this will give us a hint about the suspicion. So in case of the patient is going to die, like it will give us an idea about the etiology or cause of the death in the particular patient. Now coming to the molecular diagnosis. So you all know, like molecular diagnosis is very sensitive and the accuracy is going to be very high. So here, like we are going to subject the CSF specimen for the analysis, like where we are going to detect the 18S rRNA, which is going to be very specific for this particular infection. So depending upon the gradient, like we are going to do in the electrophoresis, like we will be having an idea about the etiological factor. So it will give us a better idea about the what is the particular etiology. In case if it is going to be an free living amoeba, it is going to tell whether it is going to be the Naegleria or Acanthamoeba or Balamutaya or Sapnia. So it will give us a better idea about the infective etiology. So again, like it is going to be a very highly sensitive and specific test for detection of these kind of invasive infection. So nowadays we do something called multiplex PCR or biofire film array, where in a single test, like we are going to detect multiple pathogens. So it is a multiplex PCR. So where we are going to detect the multiple pathogen. And moving on to the treatment. So when we study about the treatment for these free living amoeba, so unlike other infections, like we do not have a proper antiparasitic drug which is available for this particular condition. That is why the mortality rate is going to be very high. So here the mortality rate, it is going to be 98%, and there is no proper medication or antiparasitic drug which is available for this particular condition. So based upon a certain trial, so there are certain drugs like amphotericin B, rifampicin, miltefosine, which is going to have a very good result and good treatment outcome. Again, like this kind of medication can increase the this kind of medication can help the patient to come out of the particular infection. Only like 2% of the cases can come out of this particular infection. Again, 98% they are going to have a high mortality rate. So here, so among this, the amphotericin B have a very good efficacy rate. So among these three drugs, so we can go for amphotericin B for the treatment of Naegleria fowleri infection. Now moving on to the prevention. The main source of this particular infection is going to be the contaminated water, where we are going to bath in the contaminated water, where this particular trophocyte is going to come in contact with our nasal mucosa and going to involve the olfactory nerve. With the help of that olfactory nerve, it is going to reach the brain, where it is going to cause the primary amoebic meningoencephalitis. So the main prevention is going to be the avoiding bathing in this kind of water. So avoiding bathing in this kind of water which is there in the pond or river. So like we'll be able to avoid this particular pathogen coming in contact with our nasal mucosa. So thereby like we can prevent the incidence of this particular infection. So we have learnt about primary amoebic meningoencephalitis, which is caused by something called Naegleria fowleri. And there are 4 free living amoeba which is going to cause infection in the human being, and one among the particular pathogens is going to be the Naegleria fowleri, which is going to cause something called primary amoebic meningoencephalitis. So you can have a further article search and you can read your textbook for further reference. So till that, like we will catch in next class with some other diseases which is going to involve the central nervous system. So till then, Dr. Ashok signing off.